All right, I think that's enough time. So those of you here with us, thank you very much. Welcome to our spring semester student run virtual lecture series here. With us this evening, we have Chris. I'm very excited to have you here, Chris. Thank you very much. And with me, as always, is my staff. So my name is Brett McAllister. I'm the Director for Graduate Admissions for the Halmos College of Arts and Sciences here at Nova Southeastern University. With me this evening as well is two of my assistant directors, Ms. Megan Troy and Ms. D. Fei Lee, as well as my graduate recruiter, Mr. Theodore Uwe, who's with us as always as well. Those of you from Marine, you're probably very familiar with Teddy. He does all the recruitment for marine sciences, so I'm sure you've gotten many emails from him at this point and hopefully get you to come to be a student with us at this great university that we have. So Chris, thank you for being here. As I said, and those of you that are watching, thank you very much. And those of you that are going to be watching later, I know we will post this onto our YouTube channel and other social media accounts at later date once this has been fully taken care of. So first of all, introducing Chris. Chris graduated from the University of Rhode Island in 2020 with a Bachelor of Science degree in marine biology and started the MS in Marine Science program in the fall of 2020 here with us at NSU. At URI, he had a lot of exposure to lab and field work, which made him want to pursue a master's degree in this field. Here at NSU, he spent some time early on volunteering with our choral labs, which made him want to work on his thesis with Dr. Abigail Renegar. In his free time, Chris likes to spend time with friends, playing ice hockey, golfing, and also apparently likes long walks on the beach, which is perfect being in South Florida because the weather's always great and it's a great atmosphere to be down here. So Chris, thank you very much for being with us this evening. Look forward to hearing about your topic. And with that, Chris, take it away from there. And thanks once again.
All right. Thank you, Brett. I am going to share my screen. Give me 1 second. All right. Do we have it, Brett? Sorry. All right. Hello, everybody. My name is Chris Mayer. I am in my third year of the Marine Science program here at NSU, and I've had the pleasure of working with Dr. Abigail Renegar and her marine toxicology lab on my thesis project, which I will be presenting to you today. I'll begin with a little background about myself. Like Brett and I just said, I'm a candidate for a Master of Science degree with a specialty in marine biology. On top of working with Dr. Renegar on my thesis project, I have also had a job working as part of a field team on Dr. Dave Gilliam's Northern Florida Reef Tract Water Quality Project for the last 2 years. I graduated with a Bachelor of Science in Marine Biology from the University of Rhode Island in May of 2020. And while I was there, I was involved in several research projects ranging from shark population and movement dynamics to trophic ecology studies using stable isotope analysis on black sea bass and Atlantic cod. I moved to Florida from Michigan in July of 2020 and spent most of my graduate school tenure during the COVID pandemic. During this presentation, I'll give some background on plastics, pollution, and the pandemic, which will ultimately tie into my research. So when you think of plastics, common items like forks, water bottles, bags, and other containers may immediately come to mind. However, the word plastic is an umbrella term used to describe materials formed by polymers, long-chained hydrocarbon compounds of the same repetitive molecules or monomers. Polymers occur everywhere in the natural environment, from cellulose to latex, from tree sap to our own DNA. Many polymers are characterized by their chain length, molecular arrangement, and composition of carbon and hydrogen. Some, however, can contain chlorine, silicon, phosphorus, and other elements or additives. Naturally occurring polymers have been utilized by humans for centuries, and the first fully synthetic polymer was developed in 1907 by the Belgian-born American Leo Baekeland and known as polyoxybenzylmethylene glycol anhydride, or Bakelite for short. Bakelite is a thermosetting resin, meaning that it keeps its shape once molded, and it became an effective product for the electrical and automotive industries because of its extremely high resistance to heat, chemical, and electrical stimuli. Leo Baekeland's creation was the cornerstone for the synthetic polymers we use today. Synthetic polymers were not mass-produced, however, until World War II, when natural sources of latex, silk, and other materials became more difficult to obtain. The growing demand for tires was met by synthetic rubber, synthetic nylon became mass-produced for parachutes, and other polymers were used to meet other demands during such a turbulent time. Today, plastics surround almost every aspect of our lives, and the plastics industry is one of the fastest growing in the United States. Synthetic polymers are derived from crude oil, natural gas, or coal, and are categorized by their thermodynamic properties, structure, and formation method. Synthetic plastics are generally classified as olefins, which is a compound made up of a carbon and hydrogen atom with at least one pair of carbon atoms linked together by a double bond. Synthetic monomers are joined together or polymerized in two ways: addition polymerization or condensation polymerization. How a polymer is produced and how it settles or cures after it is polymerized is important in understanding its properties and application. Synthetic plastics are categorized by two main classifications: thermoplastics and thermosetting plastics, which I'll dive into now. Thermoplastics can be heated, cured, and reheated again to take on a different shape. They're easily processed or recycled, and there are no changes in their chemical properties if they get reprocessed. Thermosetting plastics, on the other hand, are generally stronger than thermoplastics because of the polymer's molecular cross-linking, which can be seen in the figure on the right. After a thermosetting plastic is cured, the plastic becomes set and can no longer be made pliable. After they're cured, thermosetting polymers will simply burn if they're exposed to high heats again. Thermosetting plastics are not only incredibly strong, but they are also highly resistant to solvents. In general, plastics that are formed by addition polymerization, such as polyethylene and polypropylene, are thermoplastic in nature, and plastics formed by condensation polymerization, such as polyester, are thermosetting in nature. Both types of plastics have their own unique applications, but their relative durability is what all synthetic polymers have in common. So like everything in this world, plastics degrade over time. And what makes the usefulness and application of modern plastics a double-edged sword is how long it takes for them to degrade once they're discarded. Some plastics can last decades, but others can take hundreds of years to fully degrade. And the property of high durability makes them a significant threat to the natural environment. Although synthetic plastics have been in production for over 100 years, they have only been recognized as an environmental concern over the last 5 or 6 decades. According to the International Union for Conservation of Nature, over 300 million tons of plastic are produced every year for a wide variety of applications. Out of those 300 million tons, 14 million tons end up in the world's oceans every year, and 80% of all marine debris throughout the water column is plastic. Different plastic polymers have different densities, which ultimately affect the area of the water column in which they reside. For example, polyethylene terephthalate, or PET, has a density of about 1.38 grams per cubic centimeter, which causes it to sink and be more prevalent in the benthic realm. Polypropylene, on the other hand, has a density of 0.905 grams per cubic centimeter and will be found in surface waters. Stormwater runoff, littering, and various industrial activities are significant sources of plastic pollution in the oceans. As one can expect, heavily populated urban centers and coastal areas, like Miami, for example, also provide a substantial amount of garbage to our shorelines. Oceanic debris has adverse effects on the well-being of marine ecosystems and the livelihood of economies that rely on the services our oceans provide. Larger marine organisms, including fish, turtles, and birds, often mistake marine debris for prey items, which can lead to starvation, lacerations, and entanglement, which eventually causes death. I'm sure we've all seen photos and videos of whales and turtles tangled in fishing nets, but to date, reports of nearly 700 aquatic organisms suffering adverse health effects due to marine debris ingestion and entanglement have surfaced. These reports range from top apex predators to zooplankton. Marine debris can also be vessels of toxicants and invasive species, including plants, protists, and fungi. Microplastics can accrue hazardous organic chemicals such as DDT, release organic contaminants such as polychlorinated biphenyls, PCBs, and polycyclic aromatic hydrocarbons, PAHs. According to Erickson et al. 2014, this problem has been recognized to the extent that some researchers have suggested classifying microplastics as hazardous waste. As plastic products break down over time, the risk of exposing living organisms at different levels of the food chain increases, and the adverse effects are still not very well understood. Economically, plastic waste can damage the aesthetic value of a tourist destination, therefore decreasing the amount of money being put into the local economy, and that adverse effect is coupled with the cost of labor and equipment used to remove garbage from a given area. Now, this brings me to the overarching topic of my research, the COVID-19 pandemic. They say that inspiration can be found anywhere, and my inspiration for this project came from being alive for the last 3 years. As we all well know, the COVID-19 pandemic was the largest public health crisis of the last century. Due to its high contagiousness and transmissibility, businesses were advised to completely shut down, and stay-at-home orders were established to stop the spread of the SARS-CoV-2 virus. Concurrently, the demand for personal protective equipment, or PPE, grew exponentially. Face coverings eventually became mandated inside of buildings and on public conveyances, such as airplanes and buses. Before the pandemic, the use of face masks was primarily only used by medical professionals in hospitals, face masks, and other PPE items. Salou et al. 2020 estimated that from January 2020 until approximately June, global surgical face mask production rose from 2.4 billion to 54 billion. At the onset of the pandemic in April of 2020, the Japanese Ministry of Economy, Trade and Industry revealed that an order of 600 million surgical face masks per month was secured. And according to Aragon 2020, China's surgical face mask production rate in June of 2020 alone was at 200 million per day. Surgical face masks are constructed by fusing a waterproof and colored outer layer, an inner layer for direct droplet absorption, and a layer in the middle that acts as a primary filtration device. Polyurethane, polyethylene, and polyester have been detected among other polymers in many types of face masks, but polypropylene is the foremost polymer found in surgical face masks. As the world dealt with the public health impacts that COVID had, the environmental repercussions began to show themselves over time. While living through the largest public health crisis of the last century, I, as I'm sure many of you, started noticing pandemic-related waste everywhere. And eventually I started asking myself some questions. So the goal of this project is to offer a unique perspective on the pandemic and its relationship to the existing plastic pollution problem. My first and most broad research question for this project is, how does a global public health crisis the size of the COVID-19 pandemic affect the natural environment? In order to dive deeper, I decided to focus on black and blue surgical face masks, two specific but very common articles of PPE that were mass-produced during the pandemic, which leads me to my second and third research questions. What magnitude does a global pandemic have on the already mounting plastic pollution problem, and at what rates do black and blue surgical face masks degrade in seawater? So why should we care? Many studies have identified polypropylene as the most abundant polymer in surgical face masks. According to Scott et al. 2022, polypropylene is the resin type that is in highest demand in Europe, and it is also the most commonly found type of plastic litter in marine and terrestrial environments. Benson, Bassi, and Palanisamy 2021 also conducted a study that calculated total face mask production by country since the start of the pandemic, and they estimated that at the time of their study, approximately 3.4 billion surgical face masks were used and discarded around the world each day. In 2019, it was estimated that about 2.3 million tons of plastic were already in the world's oceans. And as I mentioned before, plastics can take up to hundreds of years to degrade in the environment. And my experimental design aimed to simulate plastic degradation in the marine environment without actually putting more plastics into the ocean, and to assess the impact the pandemic had on this already mounting problem. So here we have a very basic overview of my experimental design. Like I just said, my goal was to simulate the marine environment in a setting that is as controllable as possible. I think of this experimental design as having liquid and solid components to it, and that will hopefully make more sense to you as I explain my procedures in further detail. It's a relatively simple design aimed at estimating degradation rates of surgical face masks over time, and I developed this procedure with the help of Dr. Renegar and my other committee members. Testing and experimentation was conducted in the Marine Toxicology Lab, Coral Histology Lab, and Sediment Lab at NSU's Halmos College of Arts and Sciences, as well as NSU's main campus in Davie, Florida. So this study took place over 16 weeks and consisted of laboratory and field exposure portions. 200 black and 200 blue for a total of 400 masks were purchased from Amazon and used in this experiment. Total of 40 2-liter Pyrex glass bottles were used, Each 2-liter bottle was filled with artificial sterilized seawater and 10 masks of the same color, as you can see in the picture on the right. 20 bottles were left inside the marine toxicology lab at ambient temperatures and away from exposure to sunlight as a control group, and 20 bottles were left outside exposed to sunlight and natural temperatures as an experimental group. Throughout the course of the experiment, each bottle was vigorously shaken once per day from Monday through Thursday to imitate wave action and provide mechanical stress to the masks inside. During this experiment, sampling took place every week for the first 4 weeks and every other week for the remainder of the experiment. On each sampling day, 1 bottle filled with black and blue masks was removed from the control and experimental groups. Each mask was removed from its respective 2-liter bottle, rinsed in 500 milliliters of fresh water, and dried overnight in a fume hood, which you can see how they looked while they were drying in the picture on the right. The seawater from the glass bottles was probed with a YSI instrument to collect temperature and salinity data, and both the seawater from the glass bottles and fresh water was used to rinse each mask was collected and filtered as part of the analysis. I'll get into the water filtration in a bit more detail a little later on, but after the masks were dried overnight, they were placed in individual 1-quart Ziploc bags for preservation and taken to the marine toxicology and coral histology labs for dry weight and light transmittance measurements. So this part of the experimental procedure is where we gathered the most simple bits of data. After the masks dried after each sampling day, they were first taken up to the coral histology lab and placed in an analytical balance, which measured their weight in grams. Weight measurements were taken before the masks were exposed to the experimental conditions. After that, they were taken back down to the marine toxicology lab, where they were placed over a sensor that measured photosynthetically active radiation, or PAR, under an LED light. The hope of this portion of the experiment was to determine whether changes in the amount of light passing through the plastic material over time could be used as a metric of degradation for face masks that are littered in the environment. This was a relatively resourceful experiment. As you can see in the picture on the right, 9 plastic beakers were stacked upside down underneath the light fixture and used as a stand for the PAR sensor. I mentioned earlier that the seawater and freshwater used in the experiment was filtered and analyzed. Each portion of water was filtered in the sediment lab with an automatic pump through an MF millipore filter paper with a 0.45 micrometer pore size and placed into a closable petri dish like the one on the right. Pictures of each filter paper were taken with a high resolution camera and are being analyzed for microfiber counts on ImageJ software. So far, counting each microfiber has been a manual process, but I'm working with the automated cell counting feature of ImageJ to save time and increase efficiency. The picture on the right is from week 1 of the experiment, by the way. After the masks were dried and weighed, light transmission measurements were taken and the water was filtered. The masks were taken to NSU's main campus in Davie, Florida for attenuated total reflectance, Fourier transform infrared spectroscopy readings. Different molecular bonds absorb different frequencies of light, and their reactions, i.e. vibrating, stretching, and bending, provide an identity for each bond and quantify the degree of degradation over time. Degradation metrics are generally displayed as indexes, and the ATR FTIR can measure the indexes of different functional groups in either solid or liquid samples. The literature states that the carbonyl index, or a carbon double bonded to oxygen, is a useful measurement for polymers such as polyethylene and polypropylene. And since surgical face masks are primarily made of polypropylene, the carbonyl index was chosen to measure degradation in the polymer's molecular functional groups. Here we can see what a blank spectrum from the FTIR looks like. This particular image was taken from my own sample readings, and it might look a little confusing, but it's actually relatively simple. The wave numbers per centimeter, or how many waves are in 1 centimeter, are on the x-axis, and the percent transmittance is represented on the y-axis, starting at 100% on the top and descending in value down the axis. Each chemical bond has a corresponding wave number range, which is documented online, and these readings can give clues as to what's happening with your particular samples. So what we can get from this particular spectrum is that there is a strong peak at approximately 1380 to 1400 wave numbers, which is shown by the blue arrow. According to LibreText chemistry, this peak could represent either a carbonyl or an alkane. The yellow arrow shows a relatively weaker peak around 1400 to 1580 wave numbers per centimeter, which represents an alkane bond. And the green arrow should show us a strong peak around 1710 to 1750 wave numbers representing a carbonyl bond. The area under the band at these peaks are ideally how we would calculate the chemical indexes of the samples that we're trying to measure. So again, the carbonyl index has been used to measure chemical oxidation of polyolefins, such as polypropylene, over time. Presence of carbonyl groups means that there's a noticeable degree of degradation in the samples, and a higher carbonyl index indicates that the samples are more degraded. Unfortunately, there is no standardized method to measure the carbonyl index. Table 3 to the right is from Almond et al. 2020, who compared and contrasted different methods of calculating the CI and proposed a method called the standard area under band, or SAUB method, as seen at the bottom of the table, to calculate it more accurately. There's no need to pay too much attention to the other method numbers on that table, but the SAUB method was chosen for this experiment in the hopes of obtaining accurate CI measurements in the masks. So you may be wondering where I'm at with my results. As of right now, I have only done preliminary data analysis, and these are what my figures look like so far, which is scary, I know. These figures represent the averages of all 10 light and weight measurements from the black and blue experimental and control groups over the 16-week experiment. It's possible that although the seawater was sterile and the masks were rinsed, there could be some degree of biofilm, salt residue, or rusting that could contribute to weight and light inconsistencies, as you can see here. There could also be variations in the mask's initial weights, but my plan is to analyze each individual mask more in depth to see if there are any noticeable trends in degradation. Obviously, my hope is that there are, and I'm hoping that the microfiber counts and CI measurements provide a more precise look at what is actually happening to the masks. So in order to finish up, there's still a fair amount to be done, although the slide says otherwise, and I'm projected to defend this project in August of this year. Mainly, I need to finish counting the microfibers on the filter papers with ImageJ, calculate the carbonyl indexes of the masks, and wrap up statistical analysis. My goal is to get this project published, so make sure to keep an eye out for that in the future. Now here we are, the end of the road. To recap, thermoplastics and thermosetting plastics are the two main types of plastics produced today, and their durability is both a blessing and a curse in modern society. Although plastics have aided in making modern life significantly more comfortable, waste has been mounting on land and at sea for decades and likely won't ever go away. The COVID-19 pandemic brought about a dramatic increase in PPE production, which, like all other waste, eventually ends up in the ocean and can have adverse effects on marine biota. Polypropylene-based plastics are among the most widespread in global pollution, and improperly disposed surgical face masks have only added to this problem. In the end, this study will provide metrics on how long it takes surgical face masks to degrade in our oceans and provide insight as to how a global pandemic can affect the natural environment. Thank you for listening. I hope you enjoyed it and ready for some questions.
Chris, thank you very much, like I said, for speaking with us this evening. That was absolutely fascinating to listen to. I really appreciate you taking some time out of your, I know, crazy busy schedule, especially you master's students, or you guys are always doing something. It's always fantastic to listen to. For those of you who have some questions, please feel free to throw it in the Q&A session, the Q&A chat box, excuse me, at the bottom. And we will go offering, and I'll read them out to you, Chris. Try and make it easy for you, make it simpler for you.
Sounds good. So Defay asked, could I explain a bit why I chose 16 weeks for the duration of the experiment? To be completely honest, this was just sort of the time frame that I had to give myself in order to get this project done in a timely manner. That's about it, to be honest. I wanted to make it long enough to provide significant results, but also I was, I'm in my third year here, and trying to graduate.
Fair enough. We have another question came through. So can you please just briefly go over your initial results again for us?
Yeah. 1 second. We'll go back. Let's see. Yeah. So if you take a look at these graphs, a lot of the results don't make a whole lot of sense. And again, these are just averages taken of each group from each week. Ideally, I would like to, with the weights, I would expect to see them go down over time. And with the light transmittance measurements, I would expect them to go up over time, which, as you can see in both of these slides and all 4 of these figures, they're kind of all over the place.
I had one I could actually ask. What would you say has been like the biggest hurdle for you so far as you did your research?
Getting started, honestly. I mulled over what I should do for my experimental design for a long time. And really, it was just all about getting the ball rolling, getting it going. And thankfully, I had a lot of help from Dr. Renegar and my other committee members. And, you know, they were able to give me the right guidance in order to get this thing moving.
And then a follow-up I would have then to that. What would you recommend for any incoming students that are looking to start that style of process? What would you recommend for them to do?
Just like getting their thesis project going?
Right, getting going, right? Like you said,
Yeah. I mean, obviously classes are important in graduate school, but start building relationships with advisors early on, start talking to them, and start just throwing ideas at them. Honestly, throw it all to the wall and see what sticks. I mean, this project for me, I was thinking about doing something with corals with Dr. Renegar, and then I just kind of had this idea, and I went to Dr. Renegar about it, and I said, "Hey, I don't know if this idea is like a little crazy, but how would you feel if I did this?" And she liked it, and so now here we are.
Awesome, thank you.
Yeah, no problem. Please try to explain, assuming you see it. DFA asked to try and explain in layperson's terms, assuming you eventually see a decent amount of degradation of the masks, would the degradation add toxicity to the ocean water? Yes. There are physical and chemical risks associated with these mass degrading seawater. You know, like a fish or a turtle, for example, mistaking mask at the surface for a jellyfish and eating it, obviously not being able to digest the plastics, causing mortality. That could be one thing. And also plastics being vessels of different, being able to carry different toxicants and different chemicals that can be harmful to life in the oceans. I think either through ingestion or exposure, you know, it could be a number of things that would be toxic for the oceans.
During your research, Chris, have you had any experience with any other crossovers, like other people doing similar research throughout the country?
I mean, not personally, but I've read a lot of read a lot of papers about it. Actually, I have another slide after this one in case anybody was interested in that. Not that one. All right, yeah, here we go. So Salou et al., which I mentioned in my presentation, they took surgical face masks and exposed and did artificial aging with them. They used artificial seawater and UV light exposures, similar to what I did. They used scanning electron microscopy to take very, very high resolution images of the actual fibers themselves breaking and fragmenting. This other one that I mentioned, this was the one that estimated total face mask production by country, and many other people such as Morgana, Casantini, and Amalfitano in 2021 have just spent time exposing surgical face masks to different shear stresses and seeing what it takes for them to break, essentially.
All right, well, Chris, if no one else has any other questions, like I said, I know how busy master students are, particularly our marine science students, with the facility and everything you guys have going on. So for those who are with us this evening, thank you very much for being here. As we mentioned before, this will be posted on our YouTube channel. So if anybody has any other context of any interest in watching this, reaching out to us, please feel free anytime to any member of my team. Or of course, we can contact you with Chris if you have further questions down the road. So Chris, thank you very much for being here. Those of you watching, I appreciate it. I hope you guys all have a wonderful rest of your Tuesday evening, and take care. Chris, thanks again.
Yeah, thank you, Brett. This was great. I really enjoyed this.
Have a good night, everyone.